Okay, so welcome to this next video in which we're discussing the alternative pathway of complement activation. Okay, right, so we've discussed so far that what's going to happen is that this complement protein C3 is continually spontaneously breaking down into C3b and C3a. Now, it's fair to say that this is a rare reaction. It does occur, but it is rare. It's not like they break down the instant they're created, okay, but there is some breakdown. Okay, now these C3b fragments then bind to glycoproteins on the surface of cells. Now they will bind to the glycoproteins on the surface of normal, healthy cells, okay, which could be very dangerous, except for the fact that normal, healthy cells have these molecules on the surface of their membranes which are there to basically bind to the C3b and stop it from triggering any further activity. Okay, so we have specialized proteins which inhibit C3b, and in addition this sialic acid molecule, also known as N-acetyl-neuraminic acid, is also found attached to other molecules on the surface of our cells, such as proteins, and it will also bind to C3b and inhibit it. Now, invading microbes don't have these protections, okay? So when C3b binds to glycoproteins on the surface of a microbe, then what will happen instead is it will now bind to another protein known as B protein, okay? And B protein will bind to C3b, and then another protein known as factor D will work on the B protein and break it down into BB and BA, okay? And BB will remain bound to C3b, whilst BA will go off, okay? Then another protein will bind, the protein properdin, and this will stabilize this complex. Then what will happen is that this complex of C3b with BB, whether it's got properdin bound to it or not, will act as a C3 convertase, and I might just jot that piece of terminology down. So a C3 convertase is just any protein complex which is capable of breaking down C3 or catalyzing the conversion of the breaking down of C3. So it will break down C3 into C3b and C3a. Okay, so you've now got this little factory set up on the surface of the microbe which is going to convert C3 into C3b and C3a. Okay, so it started off, the way it initiated is that this incredibly slow reaction led to the breakdown of C3 into C3b and C3a, and maybe just one C3b bound to the surface of the microbe. Okay, but now that binding has set up a factory which is now going to start chucking out C3b basically, and C3b will end up coating the microbe. It'll bind to glycoproteins all over the place, and it will have a positive feedback effect because all of these C3bs which have bound will also be able to recruit B protein and go through this reaction to form a C3 convertase. So you're just going to get more and more and more C3b binding to the surface of the microbe. So the microbe will get covered in glycoproteins which have got C3b bound to them. And that has the effect of opsonizing the microbe, so it will increase the chance that the microbe will be phagocytosed by neutrophils and macrophages. Okay. Right, there is another function of C3b which we will discuss in a moment after we've discussed the function of this C3a. Okay, so C3a then. So C3a is what's known as an anaphylatoxin. Okay, it can go off and activate cells in the interstitial space known as mast cells. So all over the body, stationed everywhere, you have a cell, well, a type of cell known as a mast cell. So everywhere in your body you have these cells, and they are standing by, waiting to do their job in the event of an inflammatory response. Okay? Right, so here's the nucleus of the mast cell, and basically mast cells are absolutely full of vesicles which contain histamine. Okay, so they are full of vesicles containing histamine, and these vesicles have a special name. They're often referred to as granules. Okay, so let me jot that down. Granules. And when a mast cell releases these granules, it's said to degranulate. Okay, so let me put some dots in these vesicles to denote the histamine. So this turquoise dot, these turquoise dots represent histamine molecules. Okay, so what's going to happen is that the mast cells have a receptor on their surface here, known as the C3A receptor. Okay, so I'll jot that name down here. So this is a C3A receptor. Okay, and when C3A binds to the C3A receptor on these mast cells, it triggers mast cell degranulation. So the mast cells are going to release histamine, and histamine will then go to the endothelial cells of the blood vessels in this local area. So it will go to the arterioles, the capillaries, and the venules, and it will cause type 1 activation of the endothelial cells. So it will cause more type 1 activation. I mean, we must have already had some type 1 activation, otherwise we wouldn't have got the inflammatory exudate formed, which brought the complement proteins in the first place. But it's going to cause more type 1 activation of the endothelial cells. Okay, now, C3A, as I said earlier, is known as an anaphylatoxin, and this is because of its role in anaphylactic shock. Okay, so anaphylactic shock is when let's say, for example, it's a really severe allergic reaction. It's what's incredibly dangerous about allergies. It's the potentially fatal result of a severe allergic reaction. So, for instance, if you eat something that you are really allergic to, okay, so let's say someone who's very allergic to peanuts eats a peanut, then whichever chemical it is in peanuts that triggers the allergic reaction, or whichever molecule it is, will then go into the bloodstream, and basically it will be disseminated all around the body, and it will trigger immune responses all over the body. It will trigger inflammatory responses all over the body, and what you'll get is mast cells degranulating everywhere. Okay, and when they degranulate, histamine's released into the interstitial fluid all over the body, and histamine will cause type 1 activation of endothelial cells. And type 1 activation of endothelial cells causes three things. It causes vasodilatation of the arterioles leading to that area. Okay, it causes increased permeability of the capillaries and venules in that area. Okay, so fluid moves out of the bloodstream into the interstitial space to form an inflammatory exudate. And thirdly, it leads to leukocyte extravasation. Specifically, it leads to neutrophils being recruited to the interstitial fluid. Okay, now we're not too worried about neutrophils being taken out of the bloodstream, but vasodilatation and increased permeability all over the body is not good news, because basically if that happens everywhere, then you're going to get swelling everywhere, and your blood is all just going to go into the interstitial space. Basically, all of your blood fluid will be drained into your peripheral tissues, and this is happening, remember, everywhere. Okay, so blood volume plummets, basically, really to dangerous levels, and then what happens is you get such a low blood volume that it's now not capable of performing the role that the blood is supposed to perform, which is to deliver nutrients to the peripheral tissues and to remove waste products. And when you have so little blood volume that it's no longer capable of performing the role that it was supposed to, that's known as cardiovascular failure, also known as cardiovascular collapse or anaphylactic shock. So it has a bunch of very terrifying names, and it is terrifying. It's a very dangerous disease process. And obviously, if you're not getting oxygen and glucose delivered to your peripheral tissues, then it's a matter of which dies first, the heart or the brain. So it can be fatal, basically. It's often fatal. So that's why allergic responses are so dangerous, because they can lead to anaphylactic shock. Okay, and long ago it was found that this protein C3A was involved in anaphylactic shock, so it was known as an anaphylatoxin. Okay, right, so even though this is a terrible thing in anaphylactic shock, it is helpful with regards to the normal inflammatory response, because it's going to trigger mast cell degranulation in this localized tissue. That's the difference between an inflammatory response that's local and the disseminated inflammatory response that you have in anaphylactic shock. In anaphylactic shock, it's all over the body, basically. This is a local thing where the microbe is. You want the inflammatory response to occur where the microbe is. It's going to help kill the microbe, basically. So C3A is a good thing here. Okay, right. Now, so that's another thing that the complement does. Firstly, we've got C3b coating the microbe and causing opsonization. Now we've got C3A triggering further inflammation, okay? So, now let's move on to the final thing that this pathway is going to lead to, okay? Now this involves a C3b again, okay? So let's have a look at this other function of C3b. So firstly, let's transfer our picture on here. So, we have this complex. We have the glycoprotein on the microbe membrane. We then have C3b bound to the glycoprotein here. We then have our BB fragment here, and we also have properdin bound here. Basically, we know that this is catalyzing the breakdown of C3 into C3b and C3a. Now, some of the C3b that you are producing can actually bind onto this complex. Okay, so another C3b protein can come and bind here. Okay, so let me colour this in. So we'll have the C3b in vivid purple here, and this also is a C3b protein here. And then we've got the BB in the middle, and I can't remember what colour I drew BB in, green. Okay, so BB is in green here. Okay, and then we've got properdin as well, that's just stabilising the complex and stopping it from falling apart, basically. Okay, and then we've got all of this bound to a glycoprotein on the surface of the microbe. Okay, so this complex now is known as a C3b, and then we miss out the properdin. We put BB, and then we'll put another 3b there. Okay, now what is this going to do? Well, this is actually what's known as a C5 convertase. Okay, so it's capable of breaking down the complement protein C5, so let's put our complement protein C5 here, okay, into C5a and C5b. So this entire complex now, which can form on the surface of the microbe, is going to break down complement protein 5 into a large fragment, which somehow has ended up bigger than the initial protein, but never mind, and a small fragment, which is C5a. Okay, now C5a, we'll start with this one because it's simpler, is another anaphylatoxin. Okay, so on the mast cells you also have a receptor for C5a, okay, drawn here, which is a C5a receptor. Okay, and when C5a binds to the C5a receptor on the surface of the mast cell, what this will trigger is the degranulation of the mast cell. So if this is our mast cell here, here's its nucleus, okay, then what you're going to get is the degranulation of these granules which are full of histamine, remember. Okay, so I'll put some turquoise dots in them to denote the histamine. Okay, and then when you release this histamine into the interstitial space, this will, as I said earlier, go on to cause type 1 activation of the endothelial cells. Okay, so it'll lead to type 1 activation. Okay, right. So, now, let's turn our attention to C5b. C5a is contributing along with C3a to increasing the inflammation, because it's showing, if this reaction is occurring, it's showing that there are still microbes present. So that's why you want this positive feedback of the inflammatory response, because if this reaction is occurring, it means that there are microbes in there, and therefore we need to double up our efforts, basically. Okay, so what does C5b do? Well, C5b is going to basically assemble into a complex known as the membrane attack complex. So basically it forms a complex with a whole bunch of other complement proteins. Now the first one that's going to add on is complement protein C6. Okay, and at the moment this is just forming in the interstitial space. So the first one you add on is this C6. And C6 is unlike the other complement proteins in that it doesn't need to be cleaved into two portions. Okay, so it just remains whole. So let's colour everything in so that we can keep track of it all. So what colour should we go for? We're going to have to begin the same colours over again. So C5b we'll have in red. Okay, so here is C5b. Then we'll have C6 in green. Okay. Now, so we've got the C5b C6 complex. The next one that's going to come and add on is C7, but we'll call it there for this video and continue our discussion of the assembly of the membrane attack complex in the next video.